for today, I want to focus on this topic here, plasticity and Atterberg limits. And for plasticity at Atterberg limits, we're focusing on cohesive soil, specifically clays. Okay. So we're going to talk about plasticity and Atterberg limits of clays. And so the first thing I want to discuss is just looking at different types of soils. We have discussed so far coarse grain soil, that's sands and gravel. And today we're going to focus on clays, and there's also silt. For coarse grain soils, what we know so far, first, grain size and grain size distribution matter. So for coarse grain soils, we would like to know their grain sizes and their grain size distribution and relative density, something we introduced last time. So this is an important piece of information to have for coarse grain soils. In the next chapter, we're going to talk about soil classification. And for coarse grain soils, we're going to use their grain size and grain size distribution to classify coarse grain soil. But this is very different for clays. Okay. So for clays, grain size and distribution actually are relatively unimportant. It's not that important in terms of its engineering behavior. And what's really important for clays are actually water. So water contents in clays are actually the most significant factor impacting clays behavior. Okay. So as I wrote here, clays can be categorized by the way in which they interact with water and the range of water content over which they behave like a plastic material. And we'll talk more on that today. So how clay interacts with water and how water content changes clay's behavior. And last one, silt, this is a little bit special. It's in between. So for silts, water affects its behavior, but silts have very little or no plasticity. So if you add water to silts, you're not going to observe plastic behavior as clays. So silts is kind of in between material here. So that's the coarse versus fine grain soils. And then we're going to focus on this water contents in clay, how they impact clay behavior. Okay, so I'm going to draw on this slide, this axis here. As I mentioned, water contents matter for clays. So I'm going to show on this slide, this is water content. And we'll start from the lower end. So we start from a completely dry clay. So it's very dry end. So when water content or moisture content is relatively low, the clays behave like a solid. So it's kind of a solid-like material. And then as you increase the water content in clay, so clay behaves more like a semi-solid material. And if you further increase the water content in clay, it's going to behave like a plastic material. Okay. And finally, as you keep increasing water content, adding water to clay, eventually clays are going to be like a viscous fluid. So it's like a liquid behavior. So this is increasing water content. And in between these different states, there are actually we call threshold water content. And I'm going to define all these terms more rigorously, but for now, between these states, these are basically threshold moisture content at which clay changes its behavior. And the first one, solid to semi-solid, this is called shrinkage limit. And semi-solid to plastic, that threshold water content, this is called the plastic limit. And the last one is called liquid limit. And for most natural clays in the field, the moisture content are actually between liquid and plastic limits. So most natural soil, natural clays are in this range. So most natural clays, their water contents are between plastic and liquid limit. So clays are typically a plastic material. So think of like a Play-Doh, it's more like plastic. And then, so these are basically these three, shrinkage limit, plastic limit, and liquid limit. Together, they are called the Atterberg limits. So these are basically Atterberg limits. And I'm going to define each of these limits in more detail. And also I'm going to go over the tests we use to basically obtain these limits for a given clay. So the first one of the Atterberg limits, as I mentioned, that's a shrinkage limit. And the shrinkage limit is a moisture content. So this is very important here. So all the Atterberg limits, they are water contents. Okay, so that's something you need to, if you see a number 20, 30, 50, so you need to understand these refer to moisture content at which clay changes behavior. So this is a moisture water content in percent at which the transition from solid to semi-solid state takes place. So that's a shrinkage limit, SL. And then the second one, that's a plastic limit we just introduced. Again, I want to emphasize this is moisture content, the water content or moisture content in percent at which clay changes behavior. And for plastic, clay changes from semi-solid to a plastic state. Okay. So that's plastic limit, SL or PL. And the third Atterberg limit is the liquid limit, again, moisture content. Okay. So moisture or liquid limit is from plastic to liquid state. Okay. So that's the liquid limit. And if you look at the definition, so these three limits, SL. So for a given clay, so immediately you have this relationship. So shrinkage limit is the solid to semi-solid transition moisture content. So it's actually the lowest for a given soil. And plastic and then liquid limit. Okay, so that's their relationship. So this is Atterberg limits. And Atterberg limits were originally developed actually by soil scientists. So his name is Albert Atterberg. So that's why it's called Atterberg limits. And these limits, the tests were later actually standardized by two people. You probably heard their name in first lecture, Khaled and Arthur Casagrande. So these two geotechnical engineers actually standardized Atterberg limits for geotechnical engineering applications. So the next I'm going to discuss three standard tests, Atterberg limits tests, actually developed by these two people, and they're used to determine these Atterberg limits for a given soil. The first one, this is a liquid limit test. And by the way, all these three tests except the shrinkage limit. So liquid limit and plastic limit tests are part of your lab sections. And these tests come after hydrometer analysis. So after hydrometer analysis test, so you're going to perform these liquid and plastic limits. So first for liquid limit test, the method we use is called the percussion cup method, or sometimes this is called Casagrande cup method because it was proposed by Casagrande. On the right-hand side, this picture shows the device you use for liquid limit test. In terms of how this test is conducted, so first, if you look at the top figure, so this is basically you have your soil sample. So this is clay sample. So this is your clay with certain moisture content. And this clay paste or soil paste is placed in a cup, and you see there's a groove cut at the center. So you cut this groove here with a standard grooving tool. So you have a standard grooving tool to cut this at the center of your soil paste in the cup. And then you lift that percussion cup and dropped it from a height of 10 millimeter. Again, these are standard. If you use that percussion cup device, so these are all standard. So use that crank operated cam to lift and drop your soil specimen. And as you lift and drop, that groove at the bottom is going to close. And liquid limit is defined as the moisture content required to close that groove after 25 drops, after 25 blows. Okay. So that's how you determine this liquid limit from lab test. So that's, again, moisture content required to close that standard groove after 25 blows. But in reality, okay, as you change the moisture content, it takes different numbers of blows or different number of drops to close that gap. And it's very, very difficult to get exactly 25 blows to close that. So in this test, what you typically do is you prepare multiple soil samples. Each sample has a different moisture content. And then it will require a different number of blows, different number of drops to close that gap. And then you plot the results so this is called a flow curve. So as shown here, you prepare multiple samples, perform multiple tests at varying moisture content, and you're going to get different number of blows to close that groove. So typically between 15 to 35. And then you plot these results as a flow curve. And I'll show an example of flow curve. So this is an example of a flow curve. On the x-axis, this is the number of blows called capital N, and it's plotted in log scale. Okay. In the axis is log scale. So that's a number of blows to close that groove, that 12.5 millimeter groove for that sample. And if you notice on this curve, there are 4 data points. Okay. So for each of these tests, you have a moisture content and you have one corresponding N value. So you can get these 4 data points. There are 4 tests, basically 4 soil specimens prepared. And then you feed a straight line, and this is called the flow curve. And by definition, liquid limit is a moisture content corresponding to an N value of 25. So once you have this flow curve, you start from this x-axis. So you find the 25 value. So that's N equals to 25. And then using this flow curve, you find the corresponding moisture content. So that value there, and that by definition is liquid limit. Okay. So that's how in the lab you get liquid limit for a given soil. Again, you need to perform multiple tests, prepare multiple soil samples. Okay. And the slope of this flow curve, this is called the flow index. Okay. And flow index is defined, and we call this IF. It's defined as water content 1 minus 2 divided by the log of N2 over N1. Okay, so this is just basically the slope of that curve. And so notice the order of 1 and 2. So 1 and 2 are basically 2 data points on the curve. And the order is different in the denominator because we want to get a positive slope. We want to get a positive IF value, okay. But it's basically a slope. So that's the liquid limit test. And there are other options to get that liquid limit. And so the one I just introduced, that's the standard liquid limit test. And there are other options. The first one is called the 1-point method, developed by US Army Corps of Engineers. And this 1-point method basically uses just one single data point to get LL value. So this is an empirical equation. So in the standardized test, you need multiple tests, multiple soil samples to get that value. 1-point method, you just basically do one test and then use this empirical relation to get your LL value. Okay, so that's one option. And the other option is mentioned focal method, but it's also a standardized test actually to determine liquid limit. And for this test, you use a small cone. So this is that small cone. And liquid limit is defined as the moisture content at which that small cone will penetrate a distance of 20 millimeter in 5 seconds. Okay. So that's the definition of liquid limit. Again, it's very hard to get that 20 millimeter 5 seconds value. So what you do is you prepare multiple samples at different moisture content, then fit a straight line, and then get your liquid limit. Okay, so that's focal method. And the next one is plastic limit. Okay, so this is plastic limit test, PL. So this test, this picture, a set of pictures actually shows the process of this test. And the base for this test is that soil crumbles when deformed at water content below plastic limit. So if the water content is below plastic limit, then soil tends to crumble. So it tends to observe cracks when it's deformed. And so for plastic limit, so it's basically you prepare your soil sample. Again, there's something you're going to observe in the lab test. You prepare a soil sample at different moisture content, and you roll that soil paste into threads. And plastic limit is a moisture content at which soil crumbles when rolled into threads of 3.2 millimeter in diameter. So that's the definition. That's how you get plastic limit from the lab test. And this one, it requires probably many trials, especially at the beginning, if you're not familiar with the process. And you prepare soil samples at different moisture content, try to roll it into 3.2 millimeter diameter threads and see which one crumbles. So that's your moisture content. So that's plastic limit. And then the last one, focal method actually. So I'm going to skip this. This is very similar to the liquid limit. So you can use focal method to determine plastic limit. Very similar process. The only difference is the weight of the cone is slightly higher, slightly bigger for plastic limit test. So it's a very similar process. And the last one is a shrinkage limit. So shrinkage limit, so it's shown on this slide here. So on this slide, you have moisture content and volume. So shrinkage limit, the basis of this test is that as soil loses moisture content, it's going to shrink. So that's why you see this line here. So as you decrease moisture content, the volume decreases. So that's pretty intuitive. So soil is going to shrink, but at a certain point, so that's the shrinkage limit. A further decrease of moisture content will not change the volume. So that limiting point is defined as shrinkage limit. Okay. So it's moisture content again in percent at which volume of the soil mass ceases to change. Okay. So that point is shrinkage limit. Okay. And you can see beyond this point, it's just a straight line. So meaning moisture content change does not induce any volume change. Okay. So that's shrinkage limit. And then, okay. And then this basically shows pictures of shrinkage limit test. So you have your soil sample in this dish here before drying, and then you have soil sample after drying. So you measure the mass and volume before and after. And then for shrinkage limit, there's a standard expression as shown here. You can calculate shrinkage limit. So I'm going to skip all the details here. This is something you can look in the lab manual, in the material we hand out in the lab. So these are three Atterberg limit tests.